Hello, and welcome to today's session on placebo response and placebo response mitigation in clinical trials. I'm Dr. Gary Zammit, President and CEO of Clinilabs, a contract research organization dedicated to the study of neurotherapeutics. And with me today is my friend and colleague, Dr. Steve Peroutka, MD, PhD, who is a scientific advisor to Clinilabs. Welcome, Steve.
Thanks, Gary. It's good to be here. Thanks for inviting me.
Yeah. So before we get into the details of placebos and placebo response, tell us a little bit about how you got interested in the field of psychiatry.
Well, we have sort of overlapping interests at one level. So I was in college freshman year, Psych 101, fairly undifferentiated, and I heard a famous professor from Stanford, not where I was at Cornell, gave a lecture on narcolepsy, famous doctor, Professor Dement, William Dement. And he showed a video, and I can remember sitting here and thinking, this is the most amazing thing I've ever seen. And it wasn't real. I was a dad getting upset at like a Thanksgiving dinner and falling asleep in his mashed potatoes. A guy running to first base in a softball game and falling to sleep on the way. It was over dramatized, but I really got interested in the brain and how it worked. And at that point, I was decided I wanted to be a psychiatrist. So I did a lot of psychiatry in college, volunteering at a local state hospital in Baltimore, summer jobs working at the hospital in psychiatry department, volunteering to help patients, just be like a student helper, and eventually evolved into neurology and pharmacology. But I did a lot of psychiatry in my past, and obviously a placebo response is common in both neurology and psychiatry.
And you actually started out your career at Cornell, correct, with a degree in psychology.
Neurobiology and psychology. It was a joint sort of double major.
What got you interested in placebos and placebo response rates?
Well, placebo is interesting in every disease, and there's many causes of placebo. But the bottom line is that when you do clinical drug research, the problem with the placebo is that it can mask the real effect of the drug. And it doesn't do it just by, people responding to placebo, but it does it by the variation of the responses, so the numerical. So whereas placebo should, in most diseases, change nothing, you can see placebo rates up to 100% of people responding. And that gives you what's called variance, which makes it statistically difficult to differentiate from the real drug and effect.
And as I recall from talking with you earlier, there was a book that was influential to you, and you ultimately ended up meeting and getting to know the person who wrote that
Yeah.
book.
Well, I'm going to back up 1 second to get to that point, but I just want to say that placebo historically was the basis of medicine. And since we last spoke, I looked back and they can trace the use back almost 4,000 years to Egyptian scrolls, where it says that for most diseases, the mainstays of therapy, ready, are bread or honey or beer. Those were the three main Egyptian therapeutics of the time. And that held sway until 1962 when they signed a law saying that for the first time in history here in the US, the FDA would require that drugs had to do what you said. So before that, if they were safe and they were pure, you could basically say anything you want and make a claim and ideally have some data, but it wasn't scrutinized by the FDA. So once the placebo became part of drug development, that's when it became an issue. And, across all the fields where I worked, migraine, I did a lot of work in the migraine field. There is a placebo response. However, it's also a disease that can get better on its own. Major depression is a cyclical disease. It gets worse and it gets better throughout someone's life. Is that placebo or is that a real effect of your drug? Back pain. People hurt their backs all the time. Probably the number one most common injury that someone will have in their life. Almost everybody by age 60 will have had at some point some sort of back pain. Inevitably, they will get well, but how fast do they get well versus the drug? And those are all related to placebo effects. So the book you mentioned, I was in college actually, and we had to do it in the psychology department, like a thesis, a mini major review sort of thing. And I found up about a book called Persuasion and Healing by a professor from Johns Hopkins and his physician daughter. And what they said in the book was a hypothesis that why do all different types of psychotherapy work? You can be a behaviorist, Skinnerian, a Freudian, Jungian, they have all kinds of subtypes of therapy, right? But what they showed was that there were two things that were critical to making a therapy work. And there was no drug at the time, this was just psychotherapy. And those were persuasion and healing, meaning if the therapist truly believed in what they were doing and their patient believed in them, it would work. So if you believe that behaviorism was your need and your cure and your therapist agree with you and you did behavioral therapy, you'd probably get better. But if you were skeptical about the therapist, it wasn't going to work. So
Mhm.
that had a big influence. And I did get to meet Dr. Frank, and he's a great guy, retired near retirement at the time I met him. But that book, I believe, is really true, and that's the basis of the placebo response.
Yeah. It's fascinating because in clinical medicine, if the patient gets better, whether that's a response to a drug or response to persuasion in healing or some other effect, it's a good thing because the patient has responded. Clinicians like to see that, and so do patients. But in clinical trials, it's a whole different story.
Yeah, I mean, placebo historically is a good thing. Placebo response is an even better thing because it's safe. You're not doing really anything, right? We actually in the formulary at Johns Hopkins way back, they had OB caps on the formulary. OB cap is placebo spelled backwards. So we could prescribe, they were little tiny red sugar pills, real tiny little things like breath mints. And someone's complaining of this or that, you don't want to mess up their other drugs, and so you just write them every 4 hours, give them a Nobi cap. And it would mostly quiet many people down because you'd say, this should help you sleep better or whatever their complaint was. It was, minor complaints people had. And you didn't want to change and alter the medicine. So we used to, it's rare, but we actually did do it. And you can still buy them on the internet. Look up OB caps or even placebo. You can buy placebo pills on the internet.
Hmm. Hmm. But it's fair to say that's not standard fare in clinical medicine today. Yeah.
No, no, nor should it be. But um, the point is that in a trial, those effects, that persuasion and healing, has a major impact on potentially on the outcome of your study.
Yeah, and I remember you saying that in 1962, the Kefauver Act really set the stage for the pharma industry as we know it today. Before then, companies, people didn't have to prove that their drug worked. But after 1962, they did. And that did have a profound impact on new drug approvals in the United States, didn't it?
Yeah, it dropped them. I think what the numbers are like two-thirds, down by two-thirds. So there were like 45 new products a year coming out in the late 50s and 60s, and it dropped to like 15 to 17. And it's gone up a little since then, but it's never gone up to the level because, like I said, if drugs were pure and safe, marketing claims could be made. I mean, you look at, non-medical industries and you can say you make the best car or you make the best pizza or, whatever. And nobody really confirms those facts that are being touted for the products. But in medicine, it makes sense that you should, especially when you put something in your body, be assured that it should have a positive effect or
Mhm.
could have.
Right. So proof of efficacy is required. Right.
Yes.
Well, let's change gears a bit and define some terms. What is a placebo technically, and what is a placebo response?
So placebo is something that in general is some inert substance or something that is nothing to do with the disease you're trying to help. So I mentioned bread, honey, and beer. In theory, bread is good for some things, right? If you're hypoglycemic, bread's a good thing, and honey is a good thing, and maybe even beer. But in general, a placebo is something that is not expected scientifically to have any effect on the system, right? A little red sugar pill should not affect your blood pressure or cancer or migraine. Um, but for whatever reason, it's interpreted as having a positive effect. So the placebo was a way that treaters, physicians, therapists could give something to the patient because expectations are important and be convinced that if you take this, you'll get better. So, like, I used to always joke with people, tell me they have a cold, should they get antibiotics? I said, well, for a viral infection, I can give you an antibiotic and you'll get better in 7 days. If I don't, you'll get better in a week. Yeah. I mean, that's basically a good definition of a placebo. It doesn't make any difference, right? But if I had told them, oh, if I give you this placebo, you'll get better in 3 days, it's possible in some people that that would work. They would feel better,
Mhm. Mhm.
but not really.
And I've read about this in pain studies in particular, that administering one placebo can produce a therapeutic response. And then if you double the dose and give 2 tablets of placebo, you can even enhance the therapeutic response. Is that true?
It is true, and there's another side to that, though. It turns out most diseases people love once a day doses because it's more convenient, right? By the time you get to 3 a day, every 8 hours, most people, I think close to only half can actually do that every day, 3 for say a week. But with pain, they like more doses because of the psychology that I'm hurting, I need medicine now, and I don't want to wait another day. I don't want to go to sleep tonight without another pill. So a twice a day dose of nothing can sometimes be more effective than a once a day placebo. So you're right. But again, it's all in the persuasion and the concept of this is what I need and I'm getting something for it. And that somehow translates into reports of efficacy.
Now, we're both neuroscientists, so we see one particular therapeutic area in the field of clinical drug development. Um, we see drugs being developed in psychiatry and neurology, but there are many other therapeutic areas. Is placebo response more common in CNS conditions than in other types of conditions?
It's more common in subjective conditions. So, what is pain? You can't see it. You can measure it on a 0 to 10 scale like we do, but that means completely different things to each individual, right? What your pain of a 5 is could be completely different than mine, vice versa. In some neurological diseases, Alzheimer's, there's almost no detectable placebo effect. And you can think about why that could be. Anything that's objective with blood pressure measurements, you can lower your blood pressure with a placebo. Actually, that's true. But multiple sclerosis now, there's no use of placebos because we have good drugs and it really doesn't matter any longer with multiple sclerosis what the patient says in the sense that they scan your brain, they look for lesions, and then either you have it or you don't. And there's many clinical conditions where that's what it is, right? Is your arm broken or not? They take an x-ray. And so neuroscience has suffered most, it's to be fair, most with placebo issues because of the subjectivity of all of our rating scales. It gets complicated again because it's all subjective. There's no way to quantitate pain at this point.
Yeah. Yeah. Although I must say, in the sleep field, we do see a placebo response to therapeutics. For example, in insomnia, we see quite a robust placebo response in people's ability to fall asleep or stay asleep, and that has created a challenge. I've often said that many drugs in the CNS therapeutic area don't fail because they fail to produce a difference from baseline. They fail because they don't show separation from placebo. And we've seen that over and over in so many insomnia studies. But from your perspective, how much of an impact can placebo response have on a clinical trial?
Oh, I mean, it can be very dramatic. So, in fact, I just over the weekend, there's a company called Vertex that their CEO presented data recently showing that if they looked overall in their pain study, there was no effect But if they took out the sites that had the highest placebo responses, it's a pretty dramatic difference between their drug and the rest. So when you look at the placebo rates, I mentioned earlier, there's published data in Parkinson's where, one clinical trial site, everybody responded to placebo, whereas in most of the sites, no one did. It was placebo. And so why is that? And that's one of my current major interests in how can we detect early in a study high placebo responders, which is historically what people have been trying to do. Let's figure out what patients are going to be most likely to respond. But according to my construct, as I described, it's not just the patient, it's the investigator, physician, treater who also plays maybe even a bigger role in the placebo response by
Mhm.
convincing the person that it's going to work. I know of another example of a pain study where they did an interim analysis, which is a sort of middle of the study. Let's take a look and see how we're doing kind of thing, which is legit. And they showed that their drug was working. They told everybody and the investigators, and then all of a sudden, everybody started working for the placebo too, because the investigators were excited, saying, "Now, wow, we're working on this drug, and it showed it's working." And by doing that, it ruined the study, destroyed the significance.
Yeah, I want to come back to the investigator site issue, but linger just a bit more on the impact in a clinical trial. Could it be that a placebo response in one or two or five patients, could that impact the outcome of a study, or does it take more than that?
Oh no, it takes more than that, but it's not just more, it's the variability that messes it up from a statistical level. So, we use who is football score that you win 20-10, right? But imagine you don't have 20, you have somewhere between 10 and 30 points. And, statistically, your average score is 20. And then you play a team that's average is 10, but their average is 5 to 25. You see my point. The more variable that each group of numbers is. So the 4 or 5 example, they could mess you up in a study and theoretically. I can think of one example where we had a patient who said they were doing great. They were in Australia. Funny ending to this little story, but they said their pain was 9 out of 10, which is pretty bad. But they said they were feeling great. And then when we did the statistics, that one outlying patient in a small study actually destroyed the significance. And when we ended up going back to the site, questioning the investigators, the person got mixed up. They inverted it. They thought 9 was good, 9 out of 10. But they should have said 1 because they were happy and not in any pain. But that's one example, and I'm sure there's others where a few people, if they're outliers and they really are reporting different data than everyone else, it's not so much that the averages will change that much, but the variability, which then is the statistical
Mhm,
need that you have to show statistical significance.
mhm. So just a few outliers, and the additional variability can really impact the outcome of a
Yeah.
trial. Let me ask you about development programs. I've often heard that the FDA requires two positive trials in order to approve a drug, have to show separation from placebo. But sometimes sponsors run many more than two trials in order to get those two positive.
Yep.
So placebo response is really impacting development programs as a whole, aren't they?
No, I mean, in the field of neuropathic pain, where I've done a lot of work, I know at least 2 examples where they did at least 4 to get 2 positives.
Mhm.
And in the one which I was involved in, we knew we didn't have a big signal, and that's okay. The FDA, I mean, there are drugs with a 1% difference. So, you know, eh, doing 4 studies to get your 2 positives, it can be very long and expensive.
So definitely something that pharmaceutical biotech companies, the sponsors really need to consider as they're designing their clinical trials.
There are ways to do it, I think, where you can monitor the sites, you can monitor what's happening in the trial, give the good training, do rater training, and make everything as systematic as possible and look for outliers. And that's really the bottom line is that, a pain study, whereas, in the whole study, I think the data were like 22% was the average relief of pain, placebo response, 44 sites. This is a study I was involved in. But there were these outliers. Instead of an average of 22, I think 6 of the 44 sites, so 15% of the sites, 65% placebo response, 65 compared to without them, it was 15. So it was a big difference. And the drug did work and we did get approval and all that. But, had we known about those 6 sites that were really expanding the variance of the study, that really caused problems. So that's what it is. It's a psychological problem with a statistical impact, if that makes any sense.
Mhm. And you've mentioned sites quite a few times in our conversation today. Is placebo response a problem for the site or at the site level? Is that where it originates, or is it originating in a particular type of subject? Where's
Yeah,
this
so
coming from?
that's my hypothesis. I think the powers that be in the historical past have always thought about the subject as the real problem in the placebo, that certain people were more susceptible. So they did things like run-in studies, pre-dosing, and try and, quote, weed out the people more likely to respond. But as we started talking about, I think it's a combination. It's not just the person. I think someone who's more likely to respond given a standardized message in a calm manner. The message I always like is, we don't know if this product works and we need you to help. So we need your real honest opinions, and just, think of it as the jury's out, but you're the one that's going to help decide if it's effective or not.
Mhm.
And everybody should hear that message, ideally from a video or something the same way, so that there's not a chance to say, boy, have I got the drug for you, you know, and, yeah, this one, it's working really well. And, well, because that's what you see. I mean, for someone to get 83% better in a disorder where the average treated patient gets 30% better, it's pretty unlikely
Mhm.
that, there could be all the patients in this one site happen to be incredibly susceptible. Whereas, and we've even looked like within the same town. So it's not like, people in wherever are susceptible and then the people here are not. It's the sites in general communities. I use Houston as an example. We did a study there unofficially, and we looked at 2 different sites, completely different data, in the same basic community. So it's not the people as much as the combo of the people, my hypothesis, and the investigators and the staff.
So the response could have a lot to do with the way the investigators present the study to the subject, or possibly even the enthusiasm of the investigators for
The
that particular
enthusiasm of the
study.
investigator,
Yeah.
yes, and and the talk that you alluded to earlier, there is one data going back to the 1930s from Harvard Med School, Mass General Hospital, of surgeons, one who believed in their surgical approach for gastric ulcers and one who did not believe in it, and they did the same operation on different people and got completely different results. Um, it's the placebos are powerful.
Yeah.
It,
Yeah.
it can be very, very powerful. It ran medicine for 4,000 years. It had to have something.
Yeah. Yeah. And I'm sure, just for the sake of the audience, I'm sure there are multiple things that go into generating a placebo response. It's not all uh,
Right.
on the investigator or the investigator site staff. There are probably many different things
Well,
that
no, it's two
contribute to
parties.
it.
It's the patient and it's the therapist, if you will.
Yeah. Yeah.
Those are the
So
two, right? Those are the only two touch points in the response.
so um, in order to minimize placebo response and minimize variability in data sets, um, what would be the most important things that you would advise to introduce into a clinical trial protocol to mitigate placebo response?
So my 3 suggestions are: one, we talked about make a video or some very specific written instructions that should be read so that all subjects hear the exact words about the study, what the goals are, and everybody gets the same message, right? And the second is the rater training that I know you have a lot of, your group has a lot of experience in. And the reason for that is you want to decrease the variability. You want people, to be as consistently rated on subjective scales as possible. And again, that's a statistical need for the variance, right? And that's harder than you think in a pain study. I mean, explaining to someone, what is 5 out of 10 pain mean? I mean, I had my own scale for me, I heard something, but what does it mean to somebody else? So rater training where you can try and have across the sites some communication and training of those raters so that they're rating things in a similar way. Then the third thing is the newest thing is what's called scientific surveillance,
Well,
where we
wait.
monitor.
Before we get to the scientific surveillance, I'm sorry to interrupt you. What about centralized rating? Does that have a
Yeah, no, well,
role here?
that's exactly that's the most consistent you can get, and that can work great. It doesn't always work just logistically, right, with timings and global studies, et cetera, different languages, et cetera. So it's not the panacea, but if you can do it unequivocally, and we do that often. So, for instance, with the MRI, you'll get an MRI done in a study, and it will go to a central reading place, EEGs. You get an EEG here. So you don't have different EEG readers, which is a complicated thing to do, but you'll have one group do it. And that's always a good idea. We do it with lab tests, right? You usually send the labs all to the same site to do the pharmacokinetics or for the urine testing, whatever, because you don't want 16 different labs with different techniques, with different normal ranges, et cetera. So you try to centralize everything that you can, but sometimes you can't. But if you can, do it.
Yeah. I'm glad to hear you say that, and I know Clinilabs was involved in centralized rating of subjects involved in Neurocrine's valbenazine trials, and we believe that made a tremendous difference in the ability to separate active drug from placebo, to have essentially blinded expert raters rating videos of subjects when they had their movement disorder ratings done at the investigator site.
No, and that's a good example and a great success to your team because movement disorders in particular, they're not commonly done in most doctors' offices by staff members. I mean, and they're complicated. And so I'd much rather have a small team with a lot of experience doing it. And now with technology, it's easy to do through videos and looking at things, but it's the consistency.
Mhm.
So that's
Okay.
really, I mean, when you think about it, the placebo is real, placebo effect is real. But if you add on consistency at the level of the training of the sites and messaging to the subjects, at the level of the raters, getting them the more consistent you can, the better you're going to have your data tighter.
Mhm.
And then the last one, monitoring the sites, everything's all about tightening up the
Yes.
evaluations.
Yes. Okay. And now talk to us about that third element, the analytics.
So scientific surveillance is a concept it's globally known mostly by biostatisticians, but it's something that the regulatory agencies like. And what it does is say that you can monitor the sites for different things. And we do that already. We'll see who's not enrolling patients, who's enrolling too many. I mean, I had one study where it was relatively rare disease, and one site in Eastern Europe got 28 of the 30 patients. And you have to think, that seems unusual that they were all in one spot. It was even more unusual because it was all in one week that they found all these rare patients somewhere in Eastern Europe. So, you get alerted to when sites vary. We look at protocol deviations, right? And we don't like sites that have a lot of problems, that they don't do what they're being asked to do and they don't follow the protocol. So there are ways to intervene in sites. So the idea is that if we were to start looking at not unblinded data, you look at blinded data, and in the pain example I mentioned, we expected, and I did a migraine study too like this, where we thought the response should be over 35% of people getting better. And so the placebo, we figured maybe would be 25% getting better. So we wanted at that, in that range of 30%. So you say, okay, if you have a site, we expect average blinded data of 25 placebo versus 35 drug. That's what we think. If the drug doesn't work, it drops to 25. So if any site was over 60 after X amount, it's a statistical thing. You can say, okay, after the first 5 patients go into the study, if they're all coming back at 60% or better, that's unlikely given what we know about the disorder, because half of them should be pulling it down, right? Then you can do a hold. You have to do it in advance of the study, a hold, which then has a site intervention, and you go there and just like you would for low enrollment, just like you would for protocol deviations, you'd go to the site and say, look, we're getting some data that's falling out of the statistical likelihood range. Can we talk to you about placebo issues, for example, or your raters, or some sort of intervention? But you can, if you pre-plan it, stop the site from enrolling until you figure out what they can do better.
And that's acceptable from a regulatory perspective to ratchet down activity at a site if you suspect a high rate of placebo response?
You can't do it retrospectively, obviously, but if you do it prospectively, just like protocol deviations, we stop sites, not all the time at all, hopefully not, but you can stop a site from enrolling until X prescribed intervention occurs.
Right, right. And you've also made the point to me before that if in phase 2 you see certain sites that have very high placebo response rates, those might not be sites that you'd want to carry forward into phase 3.
Right. Not only that, but they're usually high enrolling sites. That's a challenge for the sponsors. They, oh, I don't want to not use a site. So I'm a very data driven person. You would have to, explain to the sponsor of a study that this is why it's not a good idea. And I'm building a little library of examples for sponsors that they'll get it quickly. But I was involved in a diabetic neuropathy study 10 years ago or so, with a major pharma partner, and we knew it was a small signal. It was really a tiny signal, but it was a safe drug and product. And, we wanted to show it worked. And this was a fourth study in this type. So I went through the sites and we just eliminated those from further consideration and found other sites. And we did the messaging, and we did all the stuff that I mentioned, Irania, et cetera. And we did get the lowest ever placebo rate in the diabetic neuropathy study, 20%. So we didn't get it to 0 because, again, you'll never get rid of everything. But the field was, like most of the field, was 30% placebo rate. We got it down to 20, and we had a 6% signal. So that was a big difference. To go 20 to 26 versus 30 to 36 doesn't sound like a big difference, but with statistics, it is.
Mhm. Tremendous that uh, that type of action could have such an impact on outcome.
Mm-hmm. And then when you look at data post hoc, which is not FDA approved, I mean, you can do it, obviously, it's not illegal, but it's not, you can't submit it. Yeah, it makes a big difference. And I'm just surprised how much of a difference it can make.
Yeah,
Because there's the push pull of, let's take the best sites to do, the most patients, and that's great when they can do that. But you got to monitor the placebo. You
yeah.
have to monitor the endpoints and look for deviations from the placebo. I'm sorry, from the average that you expect. So most people say do like 2 standard deviations. If it's 20% placebo rate, you expect plus minus 10, then go up to 40. Anything above 40 that starts coming inconsistent, so you can put very good statistical measures. That's an algorithm, and it can be done automatically as data are entered.
Mhm. So you can gain information during a trial, after the trial is over, making decisions about entry into phase 3. Um, how often do you think sponsors share information with investigators about the placebo response rates at their particular sites? And I asked because I worked as an investigator for many years. I don't think I was ever once advised what my placebo response rate was at my location. And I would like to know because I want to make sure that I had a reasonable placebo response rate. But are sponsors more and more sharing information or not really still?
No, it's near zero is the true answer. However, each site PI, if they want it, could figure out their overall response rate because they know that they've collected the data, right? So if they wanted to know, they just had to look and say in this pain example, if I was the person getting 83% of my patients to respond to a drug that was 26 was the study average, I would be a little skeptical of myself. So you can do it, but in general, you're right. The sponsors know. So I think, the way forward would be, to work with some sponsors to say, let us have a look at the blind. We don't, you don't need to know who the sites are, but just take a look at the pattern of the
Mhm.
different sites to see if you can detect it. And thus far, everything I've looked at shows a very clear non-bell-shaped curve. It's just there's 10 to 15% of sites that are kind of off the chart, and then most of the sites are in a fairly narrow range.
I would imagine that if an investigator site was aware of an issue that, the investigators are very hardworking, the site staff are very, very hardworking, they would want to make changes to improve and bring their placebo response rate into conformity with the rest of the trial. The other question I would have for you is it, at the investigator sites, are placebo response rates kind of similar from one study to the next or one year to the next, or are they variable?
It's about 80% predictive. It's not 100, but it is high. And so in both directions. So if a site has a low placebo rate, there's about an 80% chance it will. Part of that's numbers driven because if it's only 5 people and you go from 1 to 2 out of 5, it's a big average change. But in general, it really is it's the same sites. So I was at a recent pain meeting. I asked about 10 investigators if they knew their placebo rates, and only one did.
Mm.
And that's because the individual was a PI of a study. But in general, you're right. People don't find out unless you're really writing the paper up and working with a sponsor as a PI. Most of the investigators don't know. But again, they can clearly tell from what their own data are showing.
Is there anything else you want to add about placebo response or placebo response mitigation that might be important to the audience?
Yeah, no, I think like I said, placebo can be good and it's almost undervalued now. It was the mainstay of medicine, but the idea is that a lot of time with patients, we're talking clinical medicine here, not research, but with clinical medicine, just the reassurance of a physician. And we kind of lost that in modern medicine, in my opinion. The way medicine is today is, I'm sure you had a family doctor growing up. I had one. He came to our house when we were little kids and sick. And, my parents just trusted whatever this guy said. And I probably was less medicated than maybe I should have did. But that, in a way, placebos can be really useful in modern medicine, even just calming people and reassuring them. Um, and, it's not all bad. In clinical trials, however, you don't want any placebo effects. So it's ironic that there's still a role for it, even though it messes up clinical trials.
Well, well, thank you very much for these insights. We know that placebo response and placebo response mitigation is something that's important for investigators to pay attention to, certainly for sponsors to pay attention to, because it can make a big difference in the outcomes of their clinical trials and the efficiency with which they're able to develop a new drug. They may need to run fewer trials if placebo response is really controlled. So
And
these are,
less patience, less time, less money, and bigger signals. I mean, there's
yes.
nothing sounds bad about those options.
Yeah, yeah, huge potential impact. Well, thank you so much, Steve, for these insights and for sharing them with all of us. And I hope to talk to you again soon in more detail.
Gary, thanks for inviting me. It was great talking with you, and look forward to speaking again soon.
All right. Thank you, Steve.